Our last week is ahead of us. Everybody's smiling. It's beautiful outside. Today I'm going to finish up talking about lipid metabolism, and then we only have two more topics ahead of us, one being photosynthesis and one being nitrogen metabolism. So we're not too far away. So you'll see photosynthesis in a lot of ways is like what we've talked about with respect to oxidative phosphorylation. So that shouldn't be, hopefully, too confusing for you. I've had two questions this morning, or this afternoon, about oxidative phosphorylation. So if you have questions about that, please let me know. I'll be happy to try to answer them for you. I've also had a question, will I do review sessions? And yes, I will. I will plan a review session probably sometime over the weekend, not sure. It could be Monday night. Our exam is on Wednesday at 9:30 in the morning in here, I believe. And that will be a 2-hour exam. The new material will be proportional to the coverage in the class, so that means roughly 25 to 30% of the material on the exam will be new, the rest will be old. And as I've said before, make sure you're using the old exams as study outlines is the way I recommend it, because you're going to see the topics of the exams. You're not going to see the same questions for the most part, but you will see the topics of the old exams coming up. And I'm not going to go dredge up things that I haven't asked you questions about before. So you'll see some common things that are popping up. Yes, Tessa. So the note card, good question. So the note card is something I will provide later this week. The question is, because you print something out on note cards. And so the answer is no, you cannot print anything on note cards. Everything on the note card has to be in your own handwriting. And the reason I do that is that I think that there's a real benefit that you will learn in the process of making that note card. Okay? So you can't print anything out. It's got to be, everything's got to be in your own handwriting, and you've got to turn that note card in, as I've said before. So you'll have to turn in a note card. Whether or not you want to use one, you have to turn in one that I give you, and I will give you the card later this week. Okay? Yes? You can use front back if you can write on the side, and I've seen people try to do that almost. You could do that. I had a student, this is a true story, a few years ago, who asked me if he could wear different colored glasses to the exam, and I said sure. And so he wrote a card, one version in red, one version in green on both sides, and he was able to see the red different from the green, and so he actually doubled the capacity of the card. So I have since banned that. But I thought, I gave him an A just for the creativity of it. I thought it was a very cool idea, so it was pretty cool. All right. I will, don't forget that you need to turn in your corrected exams by Wednesday at 5 p.m. And I will be available tomorrow night after class to meet as many of you as need to, because you have to meet with me as you turn those in to get any extra points back from that. So please do check with me on that. Okay. So today, most of what I'm going to talk about has to do with fats and cholesterol. And most of what I say will actually be about cholesterol because the synthesis of fat is pretty boring. And it's not very complicated at all. Fats, of course, are triacylglycerols. And if we started with phosphatidic acid, which we don't have to, but I'll explain that in a second. If we start with phosphatidic acid, it's very simple. Phosphatidic acid is a glycerol backbone. It's got 2 fatty acids, and it's got a phosphate. Okay? And if we start with that, then all we have to do is clip off the phosphate to make a diacylglycerol, and then we add a fatty acid, and we've got a fat. So that is a very easy way to make a fat. Well, how did I get to this point in the first place? Well, it started out with a glycerol phosphate is what I do. A glycerol phosphate. I put 2 fatty acids on it, and I've got phosphatidic acid. So the synthesis of fat is not at all complicated. It's very simple. The synthesis of fat overlaps with the synthesis of the glycerophospholipids because they are also made from phosphatidic acid. Okay? The synthesis of fat overlaps with the synthesis of the glycerophospholipids because it also, those also are made from phosphatidic acid. You'll see a little bit of that here. Here's phosphatidic acid. Again, get that from glycerol, phosphate, and 2 fatty acids. There's that phosphate to which can be attached other things, okay? And so we can see here the synthesis of phosphatidyl serine, and we see an intermediate. We see what I call a high energy or an activated intermediate in the process. That activated intermediate, I'm not going to expect you to know the structure or any of that sort of thing, but this activated intermediate is important, okay? Activated intermediates occur in glycerophospholipid synthesis, and the details of them we're not going to sweat, okay? But I do want you to know that CDP is involved in them. Now, CDP is, of course, cytidine diphosphate, and that means it's a nucleotide. And that means that all 4 nucleotides somehow relate to energy metabolism. They all relate to energy metabolism. Of course, ATP we talk about as being the gasoline of the cell. GTP, we said, was the energy source for protein synthesis. Okay? UDP, you might remember, was combined with glucose in the synthesis of glycogen. And that also was a high energy intermediate. And now we see CDP that's involved in the synthesis of the glycerophospholipids. Okay? This activation can occur in a couple of ways, and we're not going to sweat the ways. One is to attach the CDP to the phosphatidic acid. That's one way. The other is I can attach CDP to the molecule that's being added, and we'll see that in a second as well. So if we look here, what do we see? We have a phosphatidic acid that is being attached to one of the things being attached, in this case CDP ethanolamine, and that's simply now attached to diacylglycerol to make the phosphatide down here. So it doesn't matter whether it goes on to the small molecule or the big molecule, all right? In the synthesis of glycerophospholipids, CDP is important as an activated intermediate in that process. Yes, sir? CD as in circular dichroism, as in crazy dog, as in, what else can I say? So I probably say a lot of things, but I won't say some of them. So, well, CTP is the starting material, but you see the material that's combined is CDP, right? So the starting material is, in fact, CTP, but it's the CDP that makes the activated intermediate. Okay? Okay. And there's other examples. There's another example there, blah, blah. Okay. Sphingolipids synthesis is a little bit more complicated, but I'm going to treat that very simple also. Okay? Sphingolipids are named because they look like a compound called sphingosine. I think we've talked about that before. But the starting materials for sphingolipids are very simple also. We start with palmitoyl-CoA, which is just basically palmitic acid, and serine, the amino acid. And that's the starting material to make sphingolipids. And we can decorate those in all kinds of ways. Remember, with sphingolipids, we put on sugars and some complicated sugars and things like that. And we're not going to worry about how we do that here. But the starting materials for making them come from the palmitoyl-CoA and serine. Okay. Now, probably the more interesting synthesis actually comes from cholesterol. And cholesterol is going to at first look like, oh my God, that's big and hairy and all kinds of stuff to know. But I think you're going to see there's some organizing themes that come in the synthesis of cholesterol. The synthesis of cholesterol is a product of a metabolism we call isoprenes. I-S-O-P-R-E-N-E. An isoprene is a 5-carbon molecule. So all the cholesterol compounds are made from 5-carbon building blocks. They're all made from 5-carbon building blocks. But there's a building block that's actually smaller than the 5-carbon building block. The most important building block for this overall compound is a compound you've seen a lot of already, and that's acetyl-CoA. Acetyl-CoA. So acetyl-CoA is used to make the isoprene 5-carbon unit compounds. And then those 5-carbon unit compounds are used to make cholesterol. Well, this shows what's M and what's C. Well, if we think about the structure of an acetyl group, that's 2 carbons. One of the carbons is a methyl, and one of the carbons is a carboxyl. And this shows that the entirety, this is the structure of cholesterol, the entirety of cholesterol, or the cholesterol backbone, can be made using solely the carbons from acetyl-CoA. Solely. Okay? So again, we see, as we've seen during the term, numerous examples now where we have larger molecules that are assembled from simple building blocks. We'll see that later in the week when we talk about nucleotide metabolism. We see these great big nucleotides that are made starting with some very simple compounds like amino acids. Okay? You saw it with the sphingolipids, which started with an amino acid serine and palmitoyl-CoA. Okay? And now you see it with cholesterol, where we had the very simple building block of the acetyl group of acetyl-CoA. So what I want to do is spend a little bit of time taking you through how this process actually occurs. And I think you will see, and in fact you can see it's only several steps here. You'll see that it's fairly straightforward process. Okay, well, let's start at the top, and this is abbreviated. I'm going to come back, and in fact, I think what I want to do is instead of starting with that one, I want to start with this one right here. Yes, I do. Okay, now you may remember the ketone metabolism that I talked about last time. In ketone metabolism, we put together 2 acetyl-CoA molecules. Remember I said it was the reversal of beta oxidation. We were making ultimately a 4-carbon compound. So we took 2 acetyl-CoAs, put them together, and then we added 2 more carbons from a 3rd acetyl-CoA, and we made this intermediate known as HMG-CoA, or this long mouthful of a name here. HMG-CoA. At the time I said HMG-CoA is an intermediate in the synthesis of cholesterol. So now instead of following how HMG-CoA is used to make ketone bodies, we're going to follow how HMG-CoA is used to make cholesterol. Well, let's think about this. If I took 3 acetyl-CoAs and kept the acetyl groups, I would have a molecule that has 6 carbons, not 5 carbons. And I've told you that the isoprenes are building blocks that have 5 carbons. So that means then that the 6-carbon molecule that we make, and we go through some gyrations there, 1, 2, 3, 4, 5, 6, one of those carbons has got to be lost in order for us to make an isoprene. And no, I don't care if you know this name other than HMG-CoA. I don't care if you know this name. I don't care if you know these. Well, this enzyme you should know. Actually, I should talk about that. This enzyme is absolutely, this enzyme is absolutely critical. HMG-CoA reductase, all right? The above enzyme I don't care about. But this enzyme is really important. Okay? Why is this enzyme of all the enzymes so important? Well, this enzyme makes, okay, HMG-CoA reductase converts HMG-CoA into something else that's ultimately used to make an isoprene. This enzyme is feedback inhibited by cholesterol. It's feedback inhibited by cholesterol. Now, cholesterol, as we're going to see, is made about 25 steps later. And no, I'm not going to show you all the steps. But it's made about 25 steps later. Having an enzyme early on that measures if cholesterol is present or not and turns off the synthesis of cholesterol is a very, very important enzyme. So this is a feedback inhibited enzyme. And if any of you know people who take medication to lower their cholesterol, it's this enzyme that they target. Okay? If you know anything, anybody taking medications called statins? Okay, well, lovastatin, there's a variety of them. But what they do is that they make a compound that inhibits this enzyme. Statins inhibit this enzyme. So this enzyme is really critical in cholesterol metabolism. What we're going to see with cholesterol metabolism is that there's a lot we don't know. The metabolic process we can follow, but why a person has high cholesterol, we don't always know. Okay? Or why we hear about the stories about the person that can eat a dozen eggs for breakfast every day and they have low cholesterol. Why is that the case? Well, we have some ideas, but we don't completely know. Cholesterol turns out to be one of the most interesting compounds in all of biochemistry. Okay? Because there's something like, I want to say, I can't remember if the number's right, 9 Nobel Prizes that have been won for studying cholesterol, and we still don't know a lot about it. So it tells us there's some really important things about it. What you've learned about cholesterol so far is that cholesterol is important for the membranes of your cells. And that appears to be the primary reason that the body is making cholesterol. It's not making it because so it can be bad for you. It's making it so that it can use it. The other things that come into the way it moves in the body that determines that it's actually, in some cases, actually not good for you. All right. Here's our 6th carbon compound, right? That you don't need to know the name of. You got a 6 carbon compound made by HMG-CoA reductase. It goes through a couple of gyrations, and we're not even going to worry about the gyrations. We get to 2 building blocks that we do need to know. These are the isoprenes. So isoprene is a category of molecule. When I talk about categories, it's a category of molecule. And these 2 names are important. Isopentenyl pyrophosphate. Isopentenyl pyrophosphate, you can abbreviate it IPP. The other is dimethylallyl pyrophosphate, and you can call it DMAPP. Those are the 2 isoprenes that are used to build and make cholesterol. Well, what happened? That meant that there had to be a decarboxylation to get from here, because this guy at the top has 6 carbons, and these guys down here, if you count them, each only have 5 carbons. Note that they're pyrophosphates, and that's what a pyrophosphate looks like. They have PP on the end. Okay? Pyrophosphate. That's actually a high-energy bond is what it is. Yes, Mariah. The isoprenes have 5. So these are both isoprenes. And the decarboxylation happened right here. Look at that. We went from 6 carbons down to 5 carbons. Okay? Well, so these can be interconverted. So these go back and forth. We can make one into the other real easily. No. Yeah. But remember, we're doing this millions of times. So we've got 500,000 in this one, 500,000 in this one. But we can readily interconvert the 2 very, very easily. Yes, Marissa. DMAPP, dimethylallyl pyrophosphate. All right, you've learned the worst part of it, because the rest of it gets really simple. What happens if I combine this guy and this guy? Well, if I take something that's got 5 carbons, and I add it to something that's got 5 carbons, I suppose all of you could do the math and determine that we end up with something that's got 10 carbons. And we do. The only thing you have to know about it is the name, farnesyl pyrophosphate. And I don't have a good abbreviation for that. Farnesyl, F-A-R-N-E-S-Y-L. Farnesyl-PP, if you want to call it that. Farnesyl-PP. PP is always okay for pyrophosphate. Okay? I'm sorry, I apologize. That has 15, not 10. I screwed that up. Sorry, guys. I put the two 5s together, we get 10. That's correct. It's not farnesyl. It's called geranyl, G-E-R-A-N-Y-L. G-E-R-A-N-Y-L, pyrophosphate. When you add 5 more carbons to that, then you get farnesyl. I was wondering why that didn't look right, but that's why. So geranyl pyrophosphate has 10 carbons. Farnesyl pyrophosphate has 15 carbons. And no, I don't care if you know which ones go into which of this. You should know that IPP and DMAPP together make these, but you don't need to know which ones go into which. So farnesyl has 15. If I take two 15s and I put them together, you can start to see the math, I get something that's got 30. And that's what this guy down here at the bottom is, squalene. Squalene has 30 carbons. Now, squalene, you can see, is full of double bonds out here. And those double bonds turn out to be important because they allow us to do some rearrangement in specific ways to make molecules. Notice by twisting this guy, I start to make things that look like cholesterol. I see a bunch of electron movements, blah, blah, and I end up with closed rings. These are open rings. These are closed rings. So lanosterol forms from squalene. Lanosterol forms from squalene. L-A-N-O-S-T-E-R-O-L, lanosterol. Okay? Lanosterol has 30 carbons. We haven't added anything to it. It's just simply now with rings instead of just being open. Now you look at this guy here, and you say, well, that looks a hell of a lot like cholesterol. It should be a pretty straightforward path to get from this guy down here to cholesterol. In fact, it takes 19 steps to go from lanosterol to cholesterol. 19 steps. When I was a graduate student, I had to memorize every frigging one of those steps. And I said, when I'm a professor, I'm going to make all my students do that just to get even with the world. But I mellowed over time, and so I didn't make people do that, okay? 19 steps. It's like, where's the logic in this process? It takes a lot of energy to make cholesterol. It takes a lot of the cell's resources to make cholesterol. We know, therefore, cholesterol is important. And it's also important that we, therefore, control how much we make. If we get a lot of this, we'd like it to turn off its own synthesis, and you go, aha, HMG-CoA reductase is feedback inhibited by cholesterol, right? So you might say, oh, well then, why does anybody have any cholesterol problems? Why is cholesterol a problem? If cholesterol regulates its own synthesis, then why does anybody have any... Their voice gets higher when they say that, by the way. You notice that. Why does anybody have any difficulty with cholesterol? Okay? The reason is that cholesterol, actually, the cholesterol in our body is there in 3 ways. 1, we can make it. Bang, we make it. You've seen now how you make cholesterol starting from very simple compounds. Cholesterol is in our diet. Everybody's heard about high cholesterol foods, et cetera, et cetera. Well, that's a very active and debated area of research right now. How much of a component is diet in cholesterol? Okay? There are a lot of people now who say that diet isn't the most important thing in cholesterol. I can't say I'm convinced of that yet, but I think it is not the simple picture that people had previously with respect to saturated fat. It's probably more complicated than that. So the second way we have cholesterol in our body is by our diet. And the third way that we have cholesterol in our body is by storage. We store cholesterol. So our body is waiting for that rainy day where it has to pull out all the cholesterol that it's got stored that it can use. Okay? And then there's complicating factors. Cholesterol is recycled in our body. How do we get rid of cholesterol? We don't break cholesterol down, by the way. We poop it out. We poop it out. Our intestines have a pretty good system of pulling it back out of the poop and putting it back in the system. That's a recycling system. So that complicates the picture. One of the strategies to reducing cholesterol is to reduce, by drugs, the recycling system so that more of it poops out and you're set. Okay? So you can see that cholesterol is a really complicated picture. And I'm going to make it even more complicated for you, not to make it more complicated, but because there's some really cool things to understand about cholesterol in the body. And I think as nutritionists, as many of you are in here, understanding as much as you can about that is a really good thing, especially as people are talking to you about why do I worry about high cholesterol? How do I get high cholesterol, etc. Well, before I do that, I wanted to talk about a couple of things. One were the statins. So these were the drugs I said that were used to inhibit HMG-CoA reductase. Okay? They're used to inhibit HMG-CoA reductase. Okay? They kind of look like HMG-CoA. Okay? They also kind of look like mevalonate, which you don't need to know the structure of either one of those. And there's quite a variety of them. But in each case, they are inhibiting HMG-CoA reductase. There's quite a few drugs on the market now that are used to do just that very thing. Cholesterol is not an end product in the body. It's used for making some other things as well. One of the things that cholesterol is necessary to make in the body is something called bile acids. Bile acids. So we know that our bile is this sort of yucky-looking yellowish material that's stored in our gallbladder. Or if our gallbladder has been removed, it just is continually dripping through our intestinal system. Okay? And what bile does is it helps the body to emulsify lipids. Lipids, of course, things like fat. When you eat that giant cheeseburger, you're getting a ton of fat. Fat is not soluble in water, but you're 75% water. And that fat's got to travel from your digestive system out to a variety of cells. And the way it's got to travel is through an aqueous system known as your blood. So that means, therefore, that we have to, from the very earliest places in digestion, we have to dissolve that fat in something. Well, if I told you you got grease all over your hands and you're going to dissolve that grease so that you can wash your hands, what are you going to use? Soap, detergent, right? That's what bile acids are. They're detergents. They help to solubilize the fat in your digestive system. Okay? They have a part of them that's polar. Okay? For example, here's glycocolate. All right? Look at the polar portion, and they have a portion that's nonpolar. This can interact with the fat. This can interact with the water. Excuse me. And it can therefore make that fat be soluble in water, just like soaps and detergents will solubilize those nonpolar things so that you can wash your hands. Maria? The bile acids are part polar, that part, and part nonpolar, this part. Yep. So bile acids are made from cholesterol. Okay? You can see they look like cholesterol. You see that four-ring structure, and you'd say that looks like it's something that came from cholesterol. Okay? Bile acids are also precursors of the steroid hormones. The steroid hormones. Okay? This includes the sex hormones, male and female sex hormones, known as the estrogens and the androgens, of which testosterone is an androgen and estradiol is an estrogen. They also include the progesterones. progesterone itself is involved in the maintenance of pregnancy. A fourth category of steroid hormones known as the aldosterones. Aldosterone is a specific example. Mineralocorticoids are known as the category. Right? Mineralocorticoids. Aldosterone's a good example. And these are involved in balancing electrolytes in the body. Balancing electrolytes. You've got things like sodium and potassium that you have to balance in proper fashion in your bloodstream. Mineralocorticoids play a very important role in that process. And the fifth and last category of steroid hormones are the corticosteroids, okay? Or the glucocorticoids, as they're also called. If you want to call them glucocorticoids, that's fine. Corticosteroids or glucocorticoids. These guys are involved in inflammation processes. Inflammation and pain, okay? Pain relief, actually, okay? These guys can help reduce inflammation. They can also help reduce pain. So they're given sometimes. You hear about persons taking steroids for some swelling or something that they have, that they're probably taking something like this guy right here. Now, steroid hormones have some very serious side effects. You know from people who take the sex hormones, for example, like athletes and so forth, that overdo things, have some big problems with that. Overdoing any of this is going to be problematic. Again, all these guys come from cholesterol. And no, I don't care if you know that this comes from this, comes from this, comes from this. I will say one kind of cool thing, though, and that's right down here, okay? Right down here. I don't think I've talked about this in this class. If I have, let me know, and I'll shut up. But the difference between testosterone and estradiol is interesting. The difference is, if we look here, this is not a benzene ring. But when we look down here, this is a benzene ring. Okay? This benzene ring gets here as a result of action called an aromatase, A-R-O-M-A-T-A-S-E. An aromatase catalyzes the formation of that benzene ring. Why do I care about that? It's just another thing for me to learn and memorize. Well, the reason you care about that is that there are some cancers that are responsive to estrogens. That is, their growth is stimulated by estrogens. Okay? The presence of estrogens, even in small amounts that occur in males, the presence of estrogens can stimulate the growth of a tumor. So one of the treatments for estrogen-sensitive tumors is to give a person aromatase inhibitors so that they cannot make estrogen. The body concentration of estrogen drops to 0, and the tumor can be slowed significantly as a result of that treatment. So aromatase inhibitors block testosterone going to estradiol and consequently stop some tumors if they're sensitive to estrogens. All right. Let's talk now about the movement of cholesterol and lipids in the body. We got that big cheeseburger. We got all that fat. We got all that cholesterol. We got all those vitamins. There's fat-soluble vitamins in there as well. And they all are insoluble in water. Bile acids are very good for helping the digestive system to emulsify those and make them water-soluble. But what happens when they leave the digestive system? They've got to move through the fluids of the body to get to the liver, first of all. Not first of all, let me back up on that. With one exception, they're trying ultimately to get to the liver. Okay? But they just left the digestive system. They can pass through the intestinal wall. They can be absorbed. And when they get absorbed, the bile acid doesn't go with it. The bile acid stays behind. That means, therefore, that those lipids that are insoluble in water have got to be somehow packaged up so that they stay soluble in water. Because if they precipitate out, well, you could imagine having mixtures of things wouldn't be good for your heart, for your circulatory system, etc. So they're packaged up in complexes called lipoprotein complexes. Lipoprotein, that's one word, Lipoprotein, complexes. There are several of them. The very first one that things get packaged up in when they leave the digestive system are chylomicrons. These go from the lowest density on the top to the highest density on the bottom. These are the biggest, fluffiest things, the chylomicrons. Great, big, fluffy things just floating around, full of fat, full of cholesterol, full of fat-soluble vitamins. Okay? And you might think that, well, there's the reason people have heart attacks, because they have blockage. These things are so big and fluffy, they must be the problem. And they're not. These guys pass first, after they're absorbed, into the lymph system. The lymph system. Okay? It's an odd liquid system that flows in our body. It's not the blood. It passes from the lymph system into the bloodstream. And now you got these great big complexes. Let me get you a figure to help you orient a little bit with this. You got these great big complexes that have left the intestines. They're going into the lymph system. It's chylomicrons. They pass through the lymph system into the bloodstream. And the bloodstream, what happens? Well, they just get pumped along with the blood, right? And where do they go? They go and they hit the capillaries. And the capillaries are way too small for them. Way too small for them. So what happens? They get stuck. You can say, I'm really feeling full today because my capillaries all feel like they're stuck, right? They're stuck. Well, the sticking in the capillaries is actually part of the system of distribution. Because at the capillaries, that's where the exchange of most of these materials happens. The capillaries have various enzymes called lipases. You've already seen lipases that break down fat. And so that fat that's sitting in there in those chylomicrons is a target of those lipases. And what do the lipases do? They break down the fat into fatty acids. The cells take those fatty acids up. And the chylomicron, which was great, big, and fluffy, starts to shrink, 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 shrink. I always like to think about it as the Oreo cookie from which you start eating the middle. Only in this case, you got those double middle Oreos where they're really big, right? You're eating away all that middle. The sucker's going to start shrinking, shrinking, shrinking. The chylomicron's going to get small enough, excuse me, that it becomes what we call a chylomicron remnant. And that remnant now can pass through the capillaries. Now, I want to emphasize that not all of the Oreo middle gets eaten. Just some of the fat. The cholesterol does not get touched. The cholesterol does not get touched. It just sits there. And not all the fat gets eaten either. It just sits there. Which means you got this chylomicron remnant now that's got to go somewhere. And where does it go? Well, it goes to our favorite organ, the liver, because the liver helps us to balance everything. The liver has receptors on it that take in these chylomicron remnants. I got to do the wrong place here. This one right here takes those in. And what does the liver do with them? Well, the liver can't do much with them except say, okay, God, here's more. I wish this guy would quit eating. Okay? Quit eating. One of the reasons that people, when you get high cholesterol, one of the reasons your doctor will try to first address your high cholesterol with your diet is because you've probably been loading this guy up with all kinds of stuff. What if you ate less fat? What if you ate fewer foods that had cholesterol in them? The liver has a limited capacity, folks. You can't continue to overload it. Once you get capacity, what's the liver going to do? It's just going to pass it right out into the bloodstream. Okay? So diet is the first line of defense in lowering cholesterol. Okay? If you can do it with diet, anytime you can do something with diet and avoid taking drugs, you're better off, right? If you can do that with diet, great. If you can't do it with diet, then we go to other measures. Now, we'll not talk about those measures here. First, I want to talk about how this cholesterol system works. All we've talked about so far is diet to intestines, to capillaries, to liver. But there's more to the picture because the liver doesn't just absorb things, it also has to let things out. Remember, the liver was important for controlling blood glucose. When the body needed glucose, we had the Cori cycle where the liver made glucose and released it, right? The liver is also important in trying to maintain body needs as respect to lipids. Okay? Now, I'm going to tell you what the liver does, and then after I tell you what it does, I'm going to tell you how it does it. Because I have to start telling you what before you'll understand how. The liver is a smart organ. The liver, I'm going to say, knows that the body is needing some fat, for example, needing some cholesterol. The liver is getting a signal that I'll talk about in a minute, and the liver says, oh, wow, things are really low out there. We need to package up some things so that they can go out into the bloodstream and feed the rest of the body because the body is needing cholesterol and fat right now, for example. So the liver takes and makes another set of lipoprotein complexes. It doesn't make chylomicrons. It makes something called VLDLs, very low density lipoprotein complexes, VLDLs. And what's in those? Fat, cholesterol, fat-soluble vitamins again. And what do those VLDs do? They leave the liver, they go into the bloodstream, and they do exactly the same thing the chylomicrons did. They get stuck. And when they get stuck, the lipases come out, they start eating the contents away, and these big, fluffy VLDLs shrink to a smaller molecule that has a lighter density called intermediate density lipoprotein complexes, or IDLs. More shrinkage leads to something we call LDLs, low density lipoprotein complexes. It's the LDLs that your doctor is probably going to call bad cholesterol. Here's the process. Liver is releasing VLDLs, these go to IDLs, and these go to LDLs. Okay? LDLs are really interesting. What do they contain? Well, the lipases took out fat, right? The lipases took out fat. Lipases don't work on cholesterol. So the fat concentration decreased, which meant that as we went from here to here to here, the cholesterol concentration as a percent of the total mass increased. Okay? One of the reasons your doctor says this is bad cholesterol is it actually has the highest concentration of cholesterol. But what's going to surprise you, it's not really the cholesterol, it's the problem. I'll talk about that in a minute. All right, so we've seen this happen. Now, why is the body doing this? Well, the body's getting this thing down into a size, okay, that can be absorbed by an individual cell. It's getting it down to a size that can be absorbed by an individual cell. And that LDL can bind to a cellular receptor called the LDL receptor. And that LDL receptor grabs a hold of that thing and it internalizes it into the cell. That is how cholesterol gets into your cells from outside. Okay? There's not a specific transport protein for cholesterol. It gets in through the LDL receptor when the entire LDL gets internalized. Yes, ma'am?
So is that another way that would work? Could you inhibit that?
There are strategies for inhibiting receptors like that. They're not statins, though, because statins only work on that one enzyme. But yes, there are quite a variety of strategies people use for that. You probably wouldn't want to use that strategy because that actually lowers blood cholesterol because it's moving the LDLs out of the blood and into the cell. So you probably wouldn't want to use that as a strategy. But yes. Now, this process of moving the LDL into the cell has a long name. It's called receptor-mediated endocytosis. Oh boy, there's another big name, right? Receptor-mediated endocytosis. ENDOCYTOSIS. Receptor-mediated endocytosis. That's the way in which cholesterol gets into cells from outside. Okay. Now, how many LDLs is a cell going to take in? A cell's got a limited capacity too, right? If you've had that cheeseburger, you probably didn't have 8 cheeseburgers. You probably stopped when you had 1 or 2 because you were full. Cells get full. Cells aren't going to take LDLs forever. When cells stop taking LDLs, what happens to the LDLs? Okay? Well, they just float around the bloodstream. If nothing takes them up, they float around the bloodstream, and where do they go? They end up going back to the liver. That's why this arrow goes both ways here. LDLs go back to the liver. And the liver has an LDL receptor that grabs them and says, okay, come on back in. Now, this LDL receptor is the key to high cholesterol. The liver's LDL receptor, and by the way, this LDL receptor is different from this LDL receptor. That is, most of the cells have an LDL receptor. It's different than the liver's LDL receptor. The liver's LDL receptor is the one that's the key to high cholesterol. I'll tell you why. The liver's LDL receptor is like a little counting machine. It keeps track of how many LDLs come back in. If the liver is seeing a lot of LDLs coming back in, what does it think about the body's need for LDLs? It says the need is low because the cells aren't taking them all up. So as the LDLs go up, the liver says, okay, quit putting more out here, and you're in good shape. And if your body's doing that really well and you haven't overloaded its capacity, you can eat that dozen eggs. And your cholesterol levels are going to be fine because your liver isn't going to be putting more things out. If you've exceeded your liver's capacity, the liver says, I'm sorry, it's going back out. Question?
Yeah, so you say that the LDL receptors are different
Yes.
on the liver than they are from the cells.
Yes.
How does it differentiate between the LDL that are go back to the liver versus to the liver?
It all depends on where they bind. If they bind here, they don't make it back to the liver. If they bind here, they're obviously at the liver.
They're released from the liver, so how do they not just go right back?
Well, because the blood supply is away from the liver. So this goes out of the arterial system, comes back in the venous system. Okay? Now, I'm almost done. Okay? Now, this is a really remarkable system. We know, okay, there are people who have genetic disease called familial hypercholesterolemia. Another long name. Familial, F-A-M-I-L-I-A-L. Hyper, H-Y-P-E-R, cholesterol, C-H-O-L-E-S-T-E-R-O-L, emia, hypercholesterolemia. That's all one word, hypercholesterolemia. Okay? People with familial hypercholesterolemia have deficient LDL receptors on their liver. Okay? Deficient LDL receptors on their liver means the liver never gets to see those LDLs coming back. What do you suppose the liver thinks about what the body is needing for LDLs? It thinks that everything I put out, the body is taking, because it never gets the signal back when the LDL levels are high. People who have familial hypercholesterolemia used to die by age 10 or 12 of heart attacks and stroke because their levels of LDLs were enormous. Next time I'll tell you why LDLs are bad being high, but their levels were high like that. I taught BB450 a few years ago, and I told this story in BB450 about this thing, and this woman in the class comes up and says, "My son has that." I said, "Oh, there's no way. There's no way your son's got this," because I said, "How old is he?" She says, "He's about 10. I said, "He'd be dead." "No," she said, "I'm pretty sure that's what the doctor said he had." And she showed me a picture of this kid, and he had this thing on his knee that they had discovered that had this great big welt, and it was LDLs that had accumulated. It looked like a blister, okay? And he was having terrible time. They hauled him into the doctor and found that what's considered borderline high LDL level is about 200. This kid had a level of 700, okay? Well, the miraculous thing was that the statins had just gotten started in terms of being able to be prescribed and used to lower cholesterol levels, and they put this kid immediately onto statins, saved his life, and he's functioning perfectly well today. It's a really cool example of medicine and the obvious impact it has and the immediacy of that, okay? But because of that, that kid's walking around today. All right, I'm keeping you a little bit long there. I have a great song about cholesterol, but I'm guessing probably save it till tomorrow.